just because my connection is LTE, so I don't know if we're going to be.
I got it. I got it.
Okay, there we go.
Okay.
Should be awesome.
All right, cool. Okay.
Do you want to do full screen?
Sure.
And then I'll introduce you. There we go.
If you do this again, you can just send us an email and yeah.
Great.
Thank you.
Thank you.
Okay, everyone. Well, welcome to the first CBC seminar. We're on Zoom. There's actually 13 people with us on Zoom, and so without further ado, I'm going to introduce Ben so we can get going. So, once upon a time, Ben Langmead was a grad student here at UMD and worked on many things like Bowtie and projects that he'll be telling us about today. And since then, he has become an associate professor at Johns Hopkins University. He's won a lot of awards, like the NSF Career Award, a Sloan Research Fellowship, and the Benjamin Franklin Open Science Award. And today, he's going to be telling us about pan-genomic methods for fighting reference bias. So thanks for joining us, Ben. And
Thank you. Thank you very, very, very much. It's really, really nice to be back here on my walk. So, sorry. And over time, of course, there have been many projects that have added to our growing catalog of human genetic variation. So one example is the 1,000 Genomes Project, which is what this figure is from. But there are many more examples as well, including more recent projects like HGSVC, which surveyed structural variation, the Human Pangenome Project, the T2T Project, et cetera. So over time, it's become more and more of a sort of, it seems more and more unfortunate that the main genome that we use for many of our analyses represents only one person. It's just a single linear reference genome where we have all this variation. To say it a different way, the problem that it creates is it creates a very serious source of bias when we go to do our analyses called reference bias. We have sort of a big bucket term for all these different manifestations of bias. We call them all reference bias. So to illustrate this, let's say our job is to put sequencing reads where they belong with respect to the reference genome. The reference genome, by definition, wherever there's a polymorphism, it has the reference allele. If it has, and yet Sequencing reads come from real people who don't match the reference genome. They have some alt alleles, some non-reference alleles. So when it comes time to try to decide where to place these sequencing reads, whether or not the sequencing read, which is from our donor, has the reference or alt allele determines how difficult it is to put that read where it belongs. Because all the alt alleles create mismatches and gaps that make it more difficult for us to correctly put the read where it belongs. It tricks us either into not finding any alignment for the read or putting it in the wrong spot if we have many choices. So we're always a little bit biased against these reads that carry alt alleles. And this bias manifests in all kinds of analyses, like, for example, anything we do that is allele specific. Allele specific analyses, we're always trying to count or quantify how much evidence is coming from one haplotype or the other. And this, of course, is perfectly confounded with reference bias, because reference bias also affects how much evidence we can see from one allele or the other. So for example, if this is gene 1 and we're doing RNA sequencing, and one haplotype happens to have an alt allele, but the other does not, we're ever so slightly biased against that haplotype that has the reference allele, the alt allele. And maybe in this example, it's not such a big deal because that's just one variant, and maybe our aligner is clever enough to overcome that barrier, but maybe we've got another gene over here and it's actually got a diabolical cluster of alt alleles that completely fool the aligner, and the aligner is completely unable to align reads to that gene. And again, this is perfectly confounded with what you are trying to study in this case, which is allele-specific expression. Actually, allele-specific expression is where I first recall seeing this term reference bias in this paper from long ago now, in the year 2009, which was about this very topic, right? Allele-specific expression and why it's tricky in the presence of reference bias. There are other examples too. Some regions of the genome are hypervariable. So from person to person, it's not just a smattering of alt alleles that represent the difference between the donor and the reference. It's extremely dense clusters of really, really quite different sequence. And so this screenshot here, which is an IGV screenshot, all I'm pointing out is that in this region of the genome, you can see there's all these troughs, so to speak, in the coverage picture, because those parts of the reference genome are completely unlike the sequence of the donor genome. We just cannot recover. We cannot recover the sequence that's from the donor in those regions. Here's another example from the world of primary DNA methylation. So these are bisulfite sequencing experiments, and I'll just briefly say, this issue is one whereby if you are trying to figure out whether the CPGs in the genome are methylated or not, it would fool you if you didn't know where the CPGs were. And indeed, you can be fooled into thinking that there's an unmethylated CPG when really what you're seeing is a genetic TPG. And in this example, that's what's happening. There's actually no differential methylation here at all, but depending on whether you're using one inbred strain of mouse or the other as your reference genome, you see a signal, and the direction of the signal depends on which of the two you use as your reference. So like a particular nightmare here, right? Like not only is this completely illusory, but the direction of the signal depends on which reference you chose. Okay, so all of these problems together mean that we have an issue with reference bias. And if I had to put it in a very human biomedical centric way, I would say we would like to tackle reference bias because we want to avoid living in a world where our diagnostics and therapeutics work differentially well depending on what alleles we have and whether we're from, for example, this background or that background population. Fortunately, there have been bioinformaticians and computational people on this case for quite some time. And I'm listing here, even this is only a sparse representation of what's been done, but there's been work going back a very long time, all the way back to, I put first there this genome mapper paper because one of the very first conferences I went to, I saw that paper presented. And many, many technologies have been proposed over time that are trying to make the reference genome representation more inclusive so as to avoid the reference bias problem. And that's my focus for today. One subset of these approaches are based on the idea of a pan-genome graph. So the idea of the pan-genome graph is we don't just have one individual's genome, we have perhaps several individuals' genomes, and we do some sort of mashing together, some alignment and mashing together, so that where they are all identical or very close, we collapse things down to a single path through this graph. And where there are differences between the individuals, we allow for little forks in the road. And so in this picture here, you can see the colors basically correspond to haplotypes. the haplotypes are carving out a path through the space of possible alleles in the graph. This is one very popular idea. I like this idea very much, but I want to take the graph idea and put it aside for today, because that's not my topic. But pan-genome graphs are one popular and good way of addressing this problem. But in my own group, we sort of took a different path, or at least we started with graphs, and I'll describe very briefly one result that we got using pan-genome graphs. And then we started to notice that we could do pretty well without graphs. So let me show you what we noticed. So for example, in this paper from 2018, we looked at what would happen in a pan-genome graph if we started with no variation, so started with a linear genome, and then we prioritized all the possible variants that we could add, and then we added more and more and more and more of them, starting from high priority to low priority. And that, you can imagine, that's what's happening as we're moving from left to right on the plot that you see here. And then we asked, how well does that graph representation do at helping us to figure out the right point of origin for simulated sequencing reads? And you can imagine that's the height of the line that you're seeing on the plot. Concentrate only on the red line for now. The blue, I won't explain what the blue line is, but anyway, the red line is what I'm talking about here. And what we noticed is that we didn't have to add very many variants before we got pretty good accuracy of alignment. And furthermore, if we went too far and added too many variants, there was not just diminishing returns, but actual harm after a while to our ability to get correct alignments. The idea here being that the representation can be too ambiguous. When you start to put in very, very rare alleles, there's not much benefit because not very many people have those alleles, but you are making the entire representation more confusing. It's hard to figure out exactly where the read goes. So you're actually mostly hurting yourself after a while. And so that's one thing that we learned. And that actually drove us to think about, well, what if we started at the other end of the spectrum with linear genomes, and we saw how far we have to go toward more inclusive representations before we could do better. And this paper was a follow-up called Reference Flow, which used only linear reference genomes, but several of them, actually a total of about 6, where we had one major allele reference and then 5,000 genome superpopulation specific genomes. And we aligned in some sense to all of them, and then picked the best alignment in each case, and saw how that would do at addressing reference bias. And the basic result we eventually got to is that it did almost as well as a very good pan-genome graph-based approach, which was called VG. All of this with 6 linear reference genomes. And we were able to do this in, at the time, what was a fraction of the computational requirements of the VG tool. So this was surprising to us. First of all, what you put in the graph is a touchy question. You can put in too much. Second of all, you don't have to walk too far out from the linear side of things before you can really address a good deal of the reference bias that's present in these analyses. So, however, even this idea was pretty unsatisfying to us because this is only about 6 reference genomes we're using. If you think of the math here, imagine that there's an allele in the population and it's present at 5%, say, in the population, which I think many people would call a common genetic variant. Since there's only 6 shots on that goal, we probably don't have that variant anywhere in our data set. Right? 6 genomes is just not a lot. We probably have not covered a lot of even the common genetic variation with this representation. So the next question that we were asking ourselves is, can we go bigger? So how many of these linear reference genomes can we reasonably fit in one pan-genome representation? Not a graph, not building a pan-genome graph, but building an index over many similar reference genomes. So that's what today's storyline is all about. And I'm going to try to follow a storyline that touches on big new events in the literature. So it was mentioned that I was a PhD student here. When I was a PhD student here, we were adapting that first data structure, the FM index, to the problem of aligning sequencing reads. There were a series of advances since then, some quite interesting, that culminated in one that I want to describe today called the R index, which has sort of shifted our thinking on how to do a good job of pan-genome indexing of many human genomes. So I'm going to try to chart a path through that work that leads to some of our recent work. Hopefully also there's time for future work. And I'm going to try to touch on technical ideas along the way. And as I said to the folks at lunch today, we'll all see by the end of my talk whether this was a mistake or not. because it's actually it's gonna take me going through some intuitive examples and visual descriptions, but I'm gonna go probably too quickly to really grasp on to the underlying technical stuff. But I hope I'm leaving enough ideas and breadcrumbs behind so that you could follow up if you wanted to learn more about these individual topics. And I also have a lot of videos on YouTube that you could watch, and I'll point to those at the end too. So this is the question I want to try to ask today. How can we build small, efficient, full-text pan-genome indexes that are powerful enough to solve, for example, read classification problems? You know, a read is coming off of a nanopore sequencer. How do we quickly decide whether this is a read we're interested in or not? Okay. So the story is going to start with the BWT and the FM index. I'm sorry, my titles are getting cut off at the top there, but anyway, hopefully you can see what that says. The BWT stands for Burrows-Wheeler transform, and it's really at the core of most of the algorithms that people use to analyze sequencing data. If you were to go into the Broad Institute today and break into their machine room and log into one of their servers and then do Control-C and look at the stack trace and see what's happening in that computer, there's a good chance it's querying this kind of index that I'm about to describe. That's just so much of what goes on in bioinformatics data analysis is about indexing and about this particular kind of index. So the BWT is an index that takes the letters of our text genome, say, or pan-genome, and puts them in a new order, a special order. It's in some sense the best possible order if what you want to do later is look for substrings inside that text T. This is one way to think of it. Another way it's often pictured is like this, where you take the text, you enumerate all its cyclic rotations, you sort them, you take the last column. That's another way of saying what the Burrows-Wheeler transform is. But the short way to say it is that it just takes the letters of your text and puts them in a new order according to the alphabetical order of what comes to the right. What comes to the right in the original text. And this is the basis for tools like Bowtie and BWA. And it takes this idea and combines it with some auxiliary structures, I'll describe these in a minute, so that it can solve pattern matching problems very quickly. This ordering does two things. It allows you to solve those pattern matching problems, and relatedly, it takes the inherent repetitive structure of the string T and brings it to the surface so that it's easy to see and take advantage of. And I'll show you what I mean. So first of all, I'll show you a quick animation of what it looks like to do pattern matching using this idea of the BWT. This just gives you an idea, just a picture. Okay, so in successive steps, what we're going to do is if we have our pattern P, in this case ABA, we have our text T. You can actually just see very quickly that the pattern does occur in the text in a couple of places. That's what we're going to learn here. So in each successive step, we're going to match longer and longer suffixes of our pattern, which is to say, we're going to find which rows of that matrix start with longer and longer suffixes of our pattern P. So the first step will look like this. We've identified all the rows that start with A. Second step looks like this. We found all the rows that start with BA. Third step looks like that. And lo and behold, if we just add a little color, you can see that we found those two occurrences of ABA. Okay. I have gone very quickly through this example, right? You don't quite know what's happening with those black arrows that I just drew on the screen. They seem very important. Something's happening there. And so what I want to do on the next slide is give an accounting of exactly what computation just happened as we were walking through that previous diagram, because I just want to take a piece of it so that we can concentrate on that piece. Okay, so here's a table, and that table represents everything that we did on the previous slide, all the work. Most of it is simple. Most of it is lookups and arithmetic. Part of it is harder. This rank that you see over in the sort of left column here. That's the hard part. That's the actual computational core of what's going on when we're doing pattern matching. So that rank query is the crux of what we're doing. So I'm going to take a little time to show you what is the rank query, how did we used to handle rank queries, and then what's the more pan-genome friendly way of handling rank queries. That's what I'm going to try to get through in the next few slides. So the rank query is asking a basic question. It's asking how many times some character occurs in the string up to some position, right? So in other words, how many times a character occurs in a prefix of the string. So we denote that this way. So L.rank sub C at I is asking how many times C occurs in the string L up to but not including the offset I. Okay? So for example, if this is my string, I've gone ahead and as a red subscript, I've written in all the ranks of all the characters. So for example, this character, this A here has rank 2 because there are 2 A's to the left. So each of those is annotated with its rank. Asking that question is the computational core of what happens when we match the patterns. The data structure that we use for this, usually, is this thing called a wavelet tree. I won't go through the details of the wavelet tree. It sort of looks like this, and it's a tree, so obviously something is sort of getting split at each point in the tree. Specifically, what's being split is the alphabet. So if we have an alphabet and it's got, say, 10 characters in it, well, let's say it has 4 characters in it, because that's what we care about. Then at a split in the tree, we might split it into 2 subsets, A and C over here, G and T over here, for example. But the idea behind the tree is once we've split and split, we use these constituent bit vectors to answer questions like rank queries. We have to build some additional index data structures over those bit vectors, but generally speaking, we can answer those rank queries pretty efficiently with this tree. For example, that's a pair of bounds that are pretty easy to obtain using a suffix tree. Big O of n log sigma bit space and big O of log sigma time rank, and that's where sigma is the alphabet size. It's just an example of how we can sort of do something smarter than simply scanning, but way less memory intensive than simply memorizing all the answers. It's like a good compromise, the wavelet tree. Okay, now we want to do something better than the wavelet tree, but that takes a little bit more introduction. So the other thing that the BWT does, as I said, is it takes the internal repetitive structure of the string and brings it to the surface. So let me show you what I mean by that. So specifically what it does is it gathers together like characters, like in the sense that they have similar right context. So if characters have similar right context, they end up together in the Burrows-Wheeler transformed string. So if I give you a text like this and it's got a few occurrences of the word rectangle or something like it, then you can imagine that if you are the right context ectangle, you tend to have an R to your left, right? So if we gather everything by their right context, then those R's will come together and they'll form what we call a run in the Burrows-Wheeler transform. So there'll be a bit of the Burrows-Wheeler transform that's just the same character again and again. And that's because those 3 R's had very similar right contexts. So they came together. And the more we have internal repetitive structure to the string, like here's a pretty repetitive string. You can see it's repetitive at the word, at sort of the word level. There's actually not very many runs in that string, but as soon as we take its Burrows-Wheeler transform, we get longer runs, and you can see that we have what seems to be sort of a simplified version of that original string. And by the way, the Burrows-Wheeler transform is something you can do backwards, so you can get back to the original string if you want to. So we're not throwing it in the trash by doing the Burrows-Wheeler transform. So the more repetitive it is, the fewer and longer the runs are, which is really attractive, because if we could do something with these runs at the level of the runs, then in some sense we can deal with only the non-redundant core of our collection. You know, we don't have to deal with all the redundant stuff. We've in some sense sort of like collapsed it all away to the non-redundant core. If we do this with human genomes, here are some numbers that should intrigue us, right? So like, for example, that first row is if I have one diploid genome and I throw it in the Burrows-Wheeler transform, so I take both sets of haplotypes and throw them in the Burrows-Wheeler transform, the number of characters I put in was about 6 billion, because it was 2 haplotypes of a human genome. The number of runs in the Burrows-Wheeler transform is closer to 3.2 billion, right? It makes sense because those 2 haplotypes are very similar to each other, right? So the Burrows-Wheeler transform sort of sucked out a lot of that redundancy if you measure at the level of runs. If we go to 2, 3, 4, 5, 6 pairs of haplotypes, N goes up linearly, as expected. R goes up extremely sublinearly, very, very slowly compared to N, right? And so this is very intriguing, right? It's like R is going up with the amount of distinct sequence. Could we please have data structures that don't grow with N and instead grow with R? Because if we did, we would have a real shot at these pan-genome indexes being really, really concise. Okay, all right, so R grows very sublinearly. Well, now for a little bit more history. So if the FM index, which came out in 2000, was able to accomplish count and locate queries in something like I'm being a little hand wavy here, something like big O of n space. If you've read the papers, you know they have more complicated bounds than that, but please ignore that for now. They index these, they are able to index a text in big O of n space. Wouldn't it be nice if we could replace this with big O of r? And it was in 2005 that the first idea emerged, and it figured out how to do this, not in general, but for a specific kind of query called a count query. So let me show you that. Let's see, roughly speaking, how we're able to accomplish that. Okay, and the way we try to accomplish that is, let's start with a nice repetitive text as an example. This text is very repetitive. So if I put it through the Burrows-Wheeler transform, no surprise, I get a very runny Burrows-Wheeler transform string out, right? Nice long runs and not very many of them. So perfect, it's somehow capturing the repetitiveness of that text. And then I'm going to run length encode that string. Real simple. So in other words, if I have a run of 12 O's in a row, I'm not going to just write out all the O's. I'm going to write a little tuple that says O happened 12 times. Okay, so that's my encoding of that string right there. And we'd like to be able to do rank queries with respect to something like that thing on the bottom, hopefully, because then we're doing it in big O of R space. So the idea for how to do this, which is a tad complex, but I'm going to show visually in a few steps how it's done, is we take that run length encoded string, we encode it in a handful of new data structures, not all of which are straightforwardly going to take R space, right, O of R space, but they're all pretty simple. We've got a bit vector that's marking the beginnings of all the runs with respect to the Burrows-Wheeler transform. We've got a little compacted version of the string that's only got each of the run characters represented once. And then what we have here is basically a rearranged version of what we have here, but it's arranged by alphabet instead of by where it is with respect to the Burrows-Wheeler transform string. Handful of data structures here. And if we take those data structures, throw out the original text and just keep these, we can answer rank queries. So for example, if we want to answer this rank query, which is asking how many R's are there up to but not including offset 28, well, I'm just showing you visually one way we could compute it, which is we could scan and find that there are 6 of them. But using these data structures that I showed you on the previous slide, we can do it this way. Okay, this slide's a little too complicated. I think everyone's heart rate just went up a little bit seeing this slide, thinking I'm about to walk you through this. But instead, let me just gray out a bunch of it. And we can see, first of all, that the answer is what we hoped, right? The answer is 6, so that's good. That's what we saw from the previous slide. And then if you look at the nature of what work is going on during the computation, we've got another rank, this time on a bit vector B. We've got a select, also on that bit vector. We've got a rank on that string S, okay. And we've got a select on another bit vector. And so, did we really help ourselves here? We've still got rank and select queries. Did we manage to do this in big O of R space? It turns out that if you just take that algorithm, we've got the original one here, this is the one I showed you before, and then we've got this new extended one that's with respect to this set of data structures I just showed you. If we just sort of box up pieces of it, we'd say, okay, well, what's happening in the blue box is a so-called predecessor query on the bit vector B, and there exists a big O of R data structure that lets you do this in pretty good time. Log, log, n over R. People tend to be pretty happy when you can do log, log of anything, because log is already small and log, log is super duper small. So that's a pretty good bound there. That rank query, it turns out there's a data structure that can do this in big O of R space, log, log sigma times. Sigma is the alphabet size. And then this final pair of data structures, it turns out admits a very simple data structure, which is just a cumulative array of integers. We can do that with constant lookup in big O of R space. So we just managed to take everything in that algorithm and cram it all down into big O of R space. So that's the first benchmark result. Not new, right? I showed you this is from 2005, right? So it's not the latest, greatest result. We've known how to do this for a while. So that is the big O of R on the so-called count query. In other words, we could use that query to figure out whether and how many times a substring occurs in the text T. We still have this column here that I've called locate. If we want to know where are those occurrences, like what offsets did they occur at with respect to the text, we have more work to do. And this proposal from 2005 couldn't figure out how to do that in big O of R space. That was still an open problem. It stayed open for a long time and it was only really closed in approximately 2017, 2018, with a paper describing a new data structure called the R-index, which is my focus for the whole rest of the talk. Okay, so the R-index finally closed the locate space problem and was able to solve the locate problem also in big O of R space. This part, admittedly, I really am going to kind of skip it. I'll show you one picture. Here's the picture. That picture is from the third reference here. It uses something called the toehold lemma, which was first described in 2017, and then it was adapted for this purpose in 2018. Pardon me, I'm really just going to skip this part. Sorry about that. But anyway, this is how they were able to achieve big O of R locate queries. So when that happened, one of the authors of that R-index paper got very excited and said, bioinformaticians need to know about this. And he started emailing lots of people. And he emailed me. He was like, do you want to work on this? I said, yeah, it sounds interesting. It sounds like maybe we could use, maybe now is finally the time we can start to import more of these ideas from the theory community to improve pan-genome indexing. And so we worked together. There was another big problem to solve, which is another thing I'm sweeping under the rug today, which is how do you construct these indexes efficiently? But these folks had an idea. They had this idea called prefix-free parsing. And together we implemented this and came up with a fairly practical, and I'll show you what I mean in a moment, but fairly practical tool that's capable of indexing many human genome sequences. All right, so this is the team that worked together. So big credit to Alan Connell and Tahir Mun, but also my collaborators, Travis Christina and Giovanni. So the first set of results involved applying this tool to indexing many, in this case, we did chromosome 19 copies. So we just took a bunch of chromosome 19s from individuals from the 1,000 Genomes Project, took larger and larger sets of them, and indexed them with different approaches. We made this relatively busy and hard to look at plot, but the point of the part out here is that the R-index approach is eventually the smallest by a lot of all the approaches that are profiled. This is a logarithmic scale on the vertical scale. This is a competing compressed index that is not terribly practical, but anyway, we won eventually competing against that. And then this is Bowtie, which just completely falls off the table after a while, right? It can't, because its index is growing linearly, it has to give up much earlier in the game of trying to index this pan-genome. So this R-index was much more practical. The other thing I think is worth emphasizing is R-index came from a group of theory folks. It's not the most tuned thing in the world. Bowtie's been around for more than a decade and it's been very highly tuned. So the fact that something was able to beat Bowtie so effectively at these larger number of genomes was very promising, even if the software itself, maybe wasn't really ready for prime time. This is showing something different, which is the time required to answer locate queries against the index. Again, moving left to right, we're indexing larger and larger pan-genomes. At first, Bowtie is winning very handily, but then if we get out to larger and larger pan-genomes, eventually the R-index is able to answer these queries much more quickly. Okay? So, little pause. I don't know about the people who teach, but I often go back and look at the recordings of my teaching, and I find that at the beginning of the recording, my voice is very calm, and I'm going very slowly, and everything's fine. And then if I skip immediately to the end of the video, I'm agitated and sweating, and I look like a crazed person. So I like to put a little break in the middle here. But the good news is we've made it halfway down this path. The next few things I want to show you are, given that we now have all this progress, how can we start to apply these ideas to read classification problems using Moni and Spumoni? And then I'll talk a little bit about other, including some radically different data structures that also accomplish some of these goals. Okay, so equipped with R-index, we thought that these held particular promise for classification problems, especially where there was a real-time component, right? So for example, in nanopore sequencing, you're sequencing a single molecule in real time, and further, the nanopore has a mode called read until, where you can ask the nanopore to reject the read if you think it's uninteresting. It's a way of helping you concentrate on only the part of the sample that's most interesting to you. So this was a situation where algorithms that are able to process the data in an online fashion, unit by unit, as it's being produced, are particularly attractive. And so the group was thinking about how do we do this with R-index? What's a good online algorithm that we can construct that's built on R-index that lets us solve problems like this? And Max Rossi, who was an excellent, at the time, postdoc who was working with Christina Boucher, he's now at Illumina, he developed an algorithm called MONI, which is able to compute something called matching statistics, which is something I learned about in Mihai's class when I was a grad student here, which in turn, from matching statistics, you can also compute MEMS, or maximal exact matches, very popular kind of thing to want to compute in sequence similarity problems. So just to give you the definition here, the matching statistic of a pattern with respect to a text is the length, or the ith matching statistic, is the length of the longest prefix of the ith suffix that occurs in T. So for example, with this particular string here, look here, I've got a 5. That's a 5, because if I look at this suffix here, the longest prefix of that suffix that occurs in T is this one, because it occurs right here. So that's what that vector is at every position. So you can imagine, as my match is getting longer and longer, if you were to sweep your eyes right to left across this more visual representation, you can see it's growing as I get a nice long match, and then it comes back down as my match ends, and then it grows again as I get another nice long match, and so on. If you were to take, approximately speaking, the local maxima, those would be the MEMS, approximately speaking. Okay, so he came up with an algorithm for computing these things, and I'm going to show you a few rounds of what the algorithm looks like, so I can make a couple of points about how to turn it into an online algorithm. So here's the idea. This algorithm will start at an arbitrary point in this same matrix that we saw before. I have to show a larger example for this to be interesting. So here's a larger Burrows-Wheeler matrix. And then it's going to move character by character through the pattern right to left. And for each character, it's going to do one of two things. It's going to match, or it's going to reposition, then match. So it's got a choice. It either matches immediately or it repositions and then it matches. As we'll see, in order to reposition, it has to do some reasoning about common prefixes of rows of this matrix. So let's see an example. Let's say arbitrarily that we start in this row. We can actually start anywhere we want, believe it or not, for this algorithm. But let's just say we started in this row. We're going to take the final character of our pattern there, which is a D. We're going to hope that we see D over here in the last column, but we don't. We see $. So we have to go looking for the nearest Ds. And we can look up and we can look down. So we have a choice. We can go up or down to the nearest D. And the way we pick whether we go up or down is we examine common prefixes. So we take our current row and we ask, do I have a longer common prefix with the D above, with the row ending in D above, or the row ending in D below? And it turns out it's the row ending in D below because I have a common prefix of length 4. So that's where I'm going to jump next. I'm going to jump down. That's my repositioning step. And now I can match that D. I can go to the next step using what's called the LF mapping. I'm going to jump to the second D in the first column, and then I get to repeat this process. So I'm going to sort of wend my way slowly through these repositioning and matching steps and so on. And you can see that ever so slowly, I'm building up longer and longer matching statistics working from right to left. That's the idea. In order to know what I actually have to install into this matching statistics line, obviously I'm adding 1 some of the time, but sometimes I have to put something else in there. So for example, let me skip ahead to this step. Okay, so at this step, I'm looking over here for a D, but I don't find it. So I have to jump either up or down. This time I actually only have one choice because there's no D below, so I have to jump up. And now that I've figured out that I have to jump up, I need to figure out what number, once I follow this next edge, what number do I enter in here? Because I had a match of length 5 up until there, but when I repositioned from this row to this row, I lost some of my common prefix, right? My common prefix with the previous row was only 2. So when I extended the match by 1, I was extending a length 2 match by 1, not a length 5 match by 1. So I have to put 3 in as my next number in this matching statistics array, and so on. Right, I just want to give you the impression that this longest common prefix information is part of how we decide where to jump. It's also part of what number we have to enter into the matching statistics matrix, and so on. I just collapsed the rest of the steps in one picture like this. And lo and behold, we've got back the exact same picture that we had before, the matching statistics. Okay, so the good news about this is the matching statistics are very helpful, right? You can imagine long matching statistics are a bit like really good local alignments, right? They're kind of like good blast alignments, for example, roughly speaking. So learning that we have lots of long matching statistics is learning something important about where that read probably came from. So is this an online algorithm that we just described? Almost. I showed it as though it was, but we still have to figure out how we're going to use that, how we're going to determine the LCP information as we go, the longest common prefixes. We had to somehow figure out what were the common prefixes between the rows, and we just did it visually by looking at this picture. But the algorithm has to do it somehow. Okay, so it turns out that while they are too expensive to compute on demand and too expensive to pre-compute, the idea that Max had when he wrote MONI was we don't have to pre-compute the common prefixes per se. It is sufficient to pre-compute whether we have to jump up or down in a given situation. So if we just pre-compute whether the jump should be up or down, that's enough to get through this process correctly. And we can figure out later exactly how long those matching statistics were. Okay, so that was his idea. So this is Max's picture. And if we find ourselves, for example, at this row in this matrix, at this point in the algorithm, and we go to our threshold data structure, the threshold data structure will say, oh, if you're in this row, you have to jump down, right? In other words, the threshold, if it's somewhere in here, we would just check to see if we're above or below the threshold. Because we're below it, it says that we have to jump down. You can double check and you can see that the longest common prefix is indeed with the boundary below and not with the boundary above. Here it's a tie. So anywhere in there, it actually doesn't matter if you jump up or down. So that was the basic idea. However, even though this is a clever way of pre-computing these partial answers, and we were very excited to apply it, we eventually did apply it, to problems of real-time matching of nanopore sequencing reads, it turned out it was not online because it actually consists of two passes. In the first pass, we're using the threshold information that I just mentioned, which is what's in this block right here. We're doing, typical BWT type queries. We're using the suffix array, blah, blah, blah. We're doing typical stuff here in this first loop, but that's not good enough to determine the lengths of the matching statistics. We need a whole separate loop to determine the lengths of the matching statistics. And this was bad news, right? Because we would rather do it in just one loop, because then everything would be nice and online. So our idea was to do some violence to the MONI algorithm and get rid of one of those loops and turn it into just one and live with the repercussions. So it turned out what the main repercussion is even though we can follow the exact right path through the matrix, just like the path I showed you just now that was the correct path, we don't exactly know how long the matching statistics are. We sort of have a rough idea of how long the matching statistics are. And we hope that we can live with that. And to give you a picture of what it looks like, if these are my actual matching statistics in a particular example, what we compute is something a little bit more rudimentary called the pseudo-matching lengths. And they look like this. And often when there's a big, tall peak in the matching statistics, this example is not very big, so it's hard to see, but often when there's a big, tall peak in the matching statistics, we capture it. It's not quite as tall a peak, but we manage to capture it with these pseudo-matching lengths. And what we get in return is we get to completely jettison that second loop. We can do matching statistics, not really. We can do pseudo-matching lengths in an online fashion, okay? You can think of what these pseudo-matching lengths are as being basically the best we can do to get something like matching statistics, but without full information about the common prefixes that we were just doing by eye when we were looking at this slide before. So this is how we chose to implement it, and this was implemented in a tool that we called SPU-MONI, and the SPU part is for, I think, pseudo-match streaming, pseudo-match, I forget, but anyway, we called it SPU-MONI. And the way we apply it in practice is imagine that we are sequencing with a nanopore sequencer, we're getting sequencing reads. And our goal is to basically quickly determine, as quickly as we can, whether a read that's coming in from the nanopore does or does not originate from a pan-genome. So we have a pan-genome index, Maybe our pan-genome index is over a huge number of potential pathogens, or it's over something we're trying to exclude, like if we're doing microbiome sequencing and we want to exclude human sequences, right? So maybe we have a database of things that we want to include or exclude, but at any rate, as the data is coming in, we very quickly want to make a decision as to whether it matches the database or not. The idea is that the matching statistics, or the pseudo-matching lengths, are going to have a certain distribution. If they are long, if they are, for example, much longer than the pseudo-matching lengths we would have got if we'd had a random read, as opposed to the read that we did get, then that's a good sign that that read really does originate from one of the genomes in my database. Okay, so when the read is, for example, from human, and the pan-genome index is human, then we hope that the read itself is going to have long matching statistics, but a random version of the read, like let's say we take the read and flip it around, that would be one way to make it a random read, then we hope that the matching statistics are short. And so if that's what we see a big difference between those distributions, then that's a good sign that it did originate from the reference. If, however, there's no big difference between the two, that's a good sign that it did not really originate from our pan-genome reference. So that's the basic idea that Spumoni is applying. It's compiling both positive, we call positive matching statistics that are with respect to the database, null matching statistics that are as though the database was random or as though the read was random. So when we applied this to a real scenario of trying to distinguish reads that were from the bacterial members of a mock community, and we compared it to a Minimap-based approach, which is a pretty standard-based approach. If you were to use a tool like, for example, Readfish to solve this problem, if you've heard of Readfish, that behind the scenes is using Minimap 2, so this is sort of the standard pipeline that we're comparing to. And we find that Spumoni is able to achieve comparable accuracy to Minimap 2, and its peak memory footprint is a fraction of Minimap 2's. Its index is much, much, much smaller, and it's very substantially faster compared to Minimap 2. Okay, the main thing to notice, though, is that the pan-genome index is of a very manageable size. That's the main thing we were interested in to begin with, right? Lots and lots of bacterial genomes are in this pan-genome index. I have the number up there, around 3.5 thousand genomes are in this index, and it's of a very manageable size. Okay, I'll go quickly through this part, but just earlier this week, we posted a preprint that combined the ideas of Spumoni with a second idea, which is to apply minimizer digestion to both the reference database and the reads as they come in. This also has some other improvements which make the method more appropriate in a multi-class classification setting, not just a binary classification setting. And for more information, you can check out the paper. We've got a very similar kind of result to the one I just showed you, but there's basically like an even bigger gap between Spumoni 2 and Minimap 2. So that's just the good news here is that you can layer in these two kinds of compression, the full text index, R-index style compression, which is lossless, and the minimizer style digestion, which is lossy, and they don't undo each other, right? Like they actually do interact surprisingly well. Okay, so last thing I want to say is about an even newer development that we're also very excited about, which is that, as I showed you before, at the end of the day, when we are doing rank queries, what we tend to be doing behind the scenes is taking a bunch of bit vectors, right? The wavelet tree was just made of bit vectors, right? So I'm just imagining behind the scenes, we've got a bunch of bit vectors, and we're just sort of like touching them in bunches of places and doing lots of accesses and combining all that information so that within one iteration of the algorithm, we are probably not doing a good job of staying in a local place in memory. We're sort of going all over the place, which computer scientists are always sad to hear, right? Because it means we're going to get poor locality of reference. We're going to get lots of cache misses. This is kind of the hallmark of these approaches, is they miss in the cache with wild abandon. So they have great bounds, but they have terrible cache performance, right? So poor locality of reference, multiple bit vectors, all access multiple places, lots of cache misses. But there is a new structure. It was just proposed last year. It's very exciting, called the move structure. And what it is it's also a big O of R space pan-genome data structure, but it radically rearranges the data structure so as to basically maximize locality of reference. absolute minimum number of cache misses with this particular way of rearranging the data structure. So I only have 5 or 6 minutes. I will go extra fast through this part, but the idea is if I have a runny BWT like I have here, then the sorts of functions that we're interested in computing, like for example, the rank function that I told you before, or more specifically, the so-called LF mapping, which is just the rank plus another number, they are basically permutations, right? So if you imagine a picture of what that permutation looks like with respect to my BWT, I've drawn every permutation run as a parallelogram on this picture, and the parallelograms are wide, corresponding to the fact that the runs are long, right? So if you have a runny permutation like this, there's a data structure called the move structure that's perfect for this application. In fact, it achieves a pair of bounds that none of the R-index-based approaches have been able to achieve, which is big O of R space and constant time functions, right? Everything I showed you before was a log of something, which is real good, but it's not as good as constant. This is a very simple structure. It's really just a table, and you're moving from row to row within this table. You sometimes have a little bit of fix-up work to do, which is called fast forwarding, but you can put a constant bound on how much fast forwarding has to happen in each step, and it makes it an incredibly cache friendly way of doing things. And there's an ongoing project in the group. I wanted to put Mohsen's picture up here because I know many of you know Mohsen. He's working on a project that's trying to adapt this data structure to do things like Spumoni does, but perhaps radically faster. And certainly much more predictably, we want the inner loop to be predictably very fast. And if we can put an upper bound on how many cache misses we expect in that inner loop, that would be a great way to bound it very strongly. Okay. So I'm going to the conclusion slides now, which I hope I've left enough time. I know that we're close to 3, but I had some AV issues, so maybe you'll forgive me. So far, all the practical methods that are based on the R-index, they work pretty well, but they sure are trying to avoid issues to do with the suffix array and locate queries, because even though those have fit under our nice big O of R bound, they're still really slow, and those data structures are still, practically speaking, pretty expensive. So a lot of the methods to date, including Spumoni, some other work that I didn't talk about, it's all really trying to dance around and avoid using the suffix array and locate queries. However, in the future, we can't avoid it forever. We need locate queries because we need to be able to do things like read alignment. So one day we have to make locate queries practical. And to date, the best idea, I think, for doing this is so-called BWT tunneling, which I won't have time to describe, but if you're interested, go check out BWT tunneling. You'll have to think about graphs if you look at BWT tunneling, but it actually combines very nicely with the R-index, and I think probably represents the future of less painful locate queries. Okay, so in conclusion, when we use multiple linear references together, we can avoid reference bias in much the same way that the graph aligners avoid reference bias. We don't even need very many of the linear reference genomes, but we would love to use as many as we possibly can. More assemblies are coming. The T2D project is not the only example. There are going to be many, many more projects that produce, for example, the Human Pan-Genome Consortium has already dumped another set of 90-some haplotypes worth of human reference genomes on us. More are coming. But it's really hard to take brand new assemblies and put them in common coordinates, which is what you have to do to build a graph. Right? To build a graph, you need to do something like multiple alignment of many whole genome sequences. That's hard. They're going to resist our efforts to put them into multiple alignments together. And so we need at least some of the pan-genome methods that we develop to be methods that let linear genomes, and they are linear, they really are strings in a way, let linear genomes be linear and don't try to force them into common coordinates, especially when complex structural variation will try to defeat you if you try to do that. So, and zooming out, one thing I love about this particular field is that the theory and the practice have done a really good job of growing together. Pan-genomics in particular has been a pretty solid back and forth between papers that get published in computer science venues and papers that get published in genomics venues. It's been very fun to watch. All of these formalisms are still maturing and they could use everyone's attention. So, take a look at some of these if you're interested in finding topics to work on. And then if you're interested in a somewhat higher level overview, check out my RecomSeq talk. And then also I have a YouTube channel with playlists that you might find interesting, especially my indexing playlist covers a lot of what I talked about today. Thank you to everyone who helped me, and thank you for listening. Thanks.
Well, I know it's 3, so some people might need to wander out the classes, but I think if people can stay and have questions, now is a great time to ask them.
Yeah.
How sensitive are the runs to the indels?
Yeah, very. So the question is, how sensitive are the runs to indels? So for example, if the reads are coming from a nanopore sequencer, we know nanopore base colors make all kinds of errors, and insertions and deletions are a common kind because you can't exactly predict how quickly the DNA is moving through the pore. So unfortunately, any kind of edit, whether it's an insertion, deletion, or a mismatch, interrupts these are essentially half mems that we're measuring, right? So it interrupts the half mem. So your tower, if you had a great big tower to begin with and you have a mismatch in the middle, you end up with 2 smaller towers, right? So any kind of edit or mismatch is going to divide up these matching statistics. Which is potentially an issue. So especially if we can't do anything of a soft nature when we're computing these matching statistics, like we can't allow for those mismatches and gaps, somehow forgive them when we need to, when they're in the middle of a big long match. So some of the things we've looked at are about trying to soften the algorithm so that we can allow for the occasional mismatch and gap in the middle and not necessarily let those mems break apart, or those half mems break apart. And in general, that's difficult. So we do have to live in this environment where even just a little bit of variation does break up the signal into lots of smaller signals. Yeah.
Yeah, so I mean, like first of all, this is awesome. Like I really enjoyed this talk. My question is really super high level, which is in the very beginning you showed this graph that suggests that the more variation you include,
Yeah.
the better it gets to a point, and then you
Right.
sort of fall off a cliff.
Yes.
And then you said, but we want to be able to put as many linear
Yes.
genomes as we can. So
Yeah.
how do you think about reconciling those two?
Well, I think that the falling off a cliff that we get with graphs is kind of special to graphs. And the reason I think that is because when you put lots of variation in a graph and you don't necessarily constrain anything about how the variants co-occur on haplotypes, then that's what leads to the exponential blowup, right? That's where if you have a bunch of variants in a small window, you can take any path and that's exponential blowup. I think that's the main source of what's going wrong when you include more variants in a graph. When we are indexing individual sequences, we get to piggyback on nature's solution to this problem, which is linkage disequilibrium, right? We never allow recombinations that don't actually occur in nature, and that prunes this exponential problem to a much simpler one. Is what I'm saying correct? We don't know, because we aren't terribly facile at the moment with being able to do alignment with respect to a huge number of genomes. So like Max has work where he's working on this, but I would imagine that if you do see a diminishing returns or you see an eventual sort of like going down of your alignment performance, if it happens at all, it'll be at a point much farther to the right than the one that we saw for HiSat2. I think that's probably what we're going to see, but it's a good example of something we don't know yet. Yeah.
Just to go on record, I think you're right.
Yes, I passed my master's defense again. Yeah. Yeah.
How hard is the problem of picking which genomes to put into the linearized references?
Well, for example, when we only picked the 6, we we didn't want to be all choosy about what we put in because we didn't want people to doubt our selection. So we basically, in a way, we piggybacked on 1,000 genomes in a big way by just picking the 5 superpopulations from the 1,000 Genomes Project and making 1 genome from each of those. Okay, so was that a principled thing to do? You would think maybe a more principled thing is to put them all in some high dimensional space and try to cover the space somehow, try to pick genomes that are in some sense far apart and do a good job of representing different parts of the space. We didn't try to do that, but I think what might be interesting if you do try to do that is I'd be curious if you end up picking any non-African genomes, because there's just so much more variability among African genomes than there is among any other subpopulation which has been through a bottleneck relative to Africans, that it's not, you know, it's actually not clear. If you had to pick 10, you might pick 8 Africans and 2 non-Africans. And so we didn't attempt to solve that problem. We just used the 1,000 genome superpopulations. I didn't show it, but we did another experiment where we actually used the 1,000 genomes populations, of which there are 26. So we actually did a separate experiment with 27 genomes of major allele and all the 26 populations. We did even better with respect to the reference bias measure. But the short answer to your problem is I don't know a great way of picking them, and I'd be very curious because I think it might look very different from how we actually did pick them.
I don't know.
Yeah.
Yeah.
Like, for example, the Human Pan-Genome Project did have to solve this problem, and they did it with clustering. And if you go read their paper, they have a paragraph about the clustering they did, and it ends with a couple sentences of apology about how arbitrary their particular, clustering method is, which I'm imagining the reviewers made them put in there. But like, because that's a very hard problem to truly solve well, and therefore I don't know the answer.
I imagine if you take like a handful of genomes on a order of 10, you're still going to make this many probably.
Yes, right, yeah.
Maybe the way around is not to restrict yourself to actual genomes that have been observed, but
Yeah.
create synthetic genomes that cover what you need in large amount of maximum.
Yeah. That actually is what we did. So like, for example, we tried an experiment in that 6 genome scenario where we, instead of using superpopulation representative genomes, we made a major allele reference out of each of the superpopulations. And that did surprisingly poorly, because once we made them major allele references, they became more like each other than any given individual from those populations. So at that point, we switched to making a synthetic individual by doing a Bernoulli trial at every SNP and picking whatever we picked, so that we could, in theory, we could generate as many individuals as we wanted from the different superpopulations. But major alleles would have made our performance substantially worse. Saw another hand.
I didn't want to like give anyone another question. Is there a hybrid approach between the linearized versus the graph approach, in which you can pick more genomes for highly variable regions, like the HC complex, for example, versus just picking 7 entire genomes? Yeah,
that actually, it sounds a little bit like what our GRCH38 genome is, if you include the alt loci, right? That is one way of doing a hybrid, right? In other words, that's sort of like making a graph and then collapsing it more, more aggressively, right? There is another sort of, you mentioned hybrid approach. Another hybrid approach is you do collapse things, but you do it not beforehand, but later, which is what the tunneling idea that I briefly mentioned really is. So here we put the indexes into the genome. We don't do any collapsing ahead of time. But because so many stretches of those genomes are identical, and we'd like to save ourselves the effort of, when we're doing alignment, for example, rediscovering again and again and again the same homologous location, but in many different individuals, that's where tunneling comes in, because we can do essentially another level of collapsing, but inside the R index. Essentially what it ends up doing is making the runs shorter. So it actually doesn't change R at all, so it doesn't improve the size of the index, but it makes the runs shorter. And it means that when we go to do locate queries, we don't do as many redundant ones. But that's not the same thing as, with an eye to the underlying biology, coming up with the correct way of trying to put the genomes in a multiple alignment. We sort of don't necessarily care about doing it correctly. We care about reducing the number of locate queries when we're doing tunneling.
Well, let's thank Ben again.
Thank you. Thank you.